Almost forgot to record this. Here we go. All right, so they only do one specific reaction. We can reuse them over and over and over again before they break down. A lot of times they end in the three letters ASE, meaning like A's or ACE. So like sucrase, lactase, fructase are all pretty common enzymes. Hey, there we go. Oops. And then enzymes are kind of made up of, there's three major things that go into having an enzyme do this specific reaction. So we have the enzyme itself, which is kind of this green blobby thing on this picture. The substrate is the thing that connects to the enzyme. So this would be like whatever is going to go through the chemical reaction, whatever those initial pieces are, we call that the substrate. And then there is the active site, which is kind of the little pocket on the enzyme where the reaction is going to happen. So we'll talk a lot more about the active site in just a second. But we have the enzyme, the active site, which is the little pocket, and then the substrate, which is the molecules that the enzyme is going to react with or make the reaction happen with. So the substrate is the substance that the enzyme is going to act on. So either it's a big polymer that we're going to break down into little monomers, or whether it's a bunch of little monomers that we're going to take and put together to make a big polymer. Whatever your starting material is, we just kind of give it the general name of the substrate. So the substrate is the substance that is going to go through the reaction. So the substrate is going to go into the active site on the enzyme. So that is kind of what the active site is there for. The little pocket, the little hole on the enzyme, that is where our substrate is going to go into. So we have our big enzyme protein, and then we have our substance that the enzyme is going to catalyze, is going to make go faster. So an active site is this kind of pocket region or this open region of the enzyme. Oh, yeah, sure thing, Victoria. So we have our enzyme, we have our substrate, which is going to go into the enzyme. And the place where the substrate joins, we're going to talk about in a second, is called the active site. So that's kind of this little pocket opening right here. You good, Victoria? Sweet. All right, so like I was saying, the substrate is going to go into the enzyme at a place called the active site. So the active site, like I said, is this little pocket, this little opening on the enzyme right here. And that is where your substrate is going to go into. And if you've got the correct enzyme and the correct substrate, then the substrate should fit pretty nice and snug right into the active site. Oops. So the active site and the substrate should fit together pretty perfectly. And this is where that actual chemical reaction is going to happen. So the substrate comes in here, the enzyme lowers that activation energy, and then the reaction happens. The chemical reaction goes on. So that's sort of the first part of what happens in order for these reactions to go down. In order for an enzyme to work, the first thing that's got to happen is we have to have a substrate slide right on in to the active site of our enzyme protein. So there are a couple different ways that scientists explain how the active site and the substrate kind of fit together. One of the explanations that they've come up with is called the lock and key hypothesis. And this is pretty much exactly what it sounds like. Some scientists think that the substrate and the active site should be like a perfect close fit to each other, just like a key fitting into a lock. So just like how hopefully only your house key fits into the lock on your house, and other keys, even though they might be really close, they just don't quite fit in the same way. That's one way that scientists think active sites and substrates fit together, is that they'll only go together if they are this perfect fit. Because remember, enzymes are specific for just one type of reaction. So an enzyme will only fit one substrate. An enzyme and its active site should only be able to bind to one substrate. So the way that they make sure it's the right one is by having this kind of perfect fit. So when both of them are together, scientists call it the enzyme substrate complex, because now they're all together and that reaction is going on inside the active site. The way that stuff leaves from the active site, because remember, the enzyme is reusable. So eventually we're going to have to open up that active site again so that more substrates can come in. So usually after the reaction happens, after we've catalyzed that chemical reaction, the products, the stuff that we end up with, are a little bit different shape than the original substrate. So because they're a little bit different shape, they don't exactly fit the active site, so they are released. They're let go from the enzyme. That way, the active site can be cleared out and we can get a new substrate in there. Let's get if I have, yeah, there we go. So here's kind of a diagram showing what's going on. We have our substrate, which fits perfectly into the active site of our enzyme. So they come together. Thanks to that low activation energy, the enzyme's able to get going really quick. And it turns this substrate, the starting material, into products, our ending material. So that's what the P down here stands for, is product. So like it shows on this picture, the product is a little bit different shape than the substrate. So it doesn't quite exactly fit the active site. So the products get let go. They go do whatever they're going to go do in the cell. And now the active site on the enzyme is open, and we can use it again to go through this reaction again and again and again. Substrate comes in, binds, is changed, is released. We can bring in a new substrate, bind, change, release, bring in a new substrate over and over and over again. So because these enzymes are reusable, we can use them right away after they're done with the first reaction. And because they make those reactions happen so quickly, we can really quickly move through a large amount of substrate. So again, lock and key hypothesis is just the idea that the substrate and the active site on the enzyme are like a perfect fit for one another. They fit exactly on there. It helps make sure that the right substrate gets in there, because if the wrong substrate does, then it's the wrong shape and nothing's going to happen. The enzyme will just let it go and wait for the right substrate to come by. There's one other kind of hypothesis or idea that scientists have about how substrates and enzymes work. It's called the Induced Fit Hypothesis. And it's the idea of the substrate and the enzyme and active site aren't an exact fit at first, but when the substrate comes in and binds to that active site, it changes the shape of the enzyme a little bit, and that's what causes the chemical reaction to start happening. So in some cases, maybe it's a lock and key model where the substrate comes in and binds perfectly. In other cases, it might be this induced fit idea where the substrate changes the shape of the enzyme slightly. So either way, we end up with a really snug fit between the enzyme and the substrate. The enzyme causes that reaction to happen really quickly, and then whatever our products are, whatever we make at the end of the reaction, the enzyme lets those go and is ready to be reused again for another reaction. So enzymes can only do one specific reaction, which is why there are hundreds of different enzymes in our bodies, because they're responsible for all sorts of different reactions that we have to deal with. So there's tons and tons and tons of different enzymes, and each enzyme does one reaction over and over and over and over and over again. All right, does anybody have any questions about enzymes, substrates, active sites? Any questions about anything like that before we kind of move on to the next part? All right, I'm not hearing any questions up here. So we've got our enzyme, we've got our substrate, we've got our active site. We have that nice fit between the enzyme and the substrate. And remember, if the active site doesn't fit the substrate, then that reaction isn't going to happen. We have to have the correct substrate go and fit right in. What that also means is, remember, we talked about when we went over proteins with the macromolecules, I mentioned that structure and function of proteins go right together, hand in hand. The structure of a protein is super, super important for how it works, what its function is. What that means is especially true for enzymes. If the shape of the active site isn't exactly right for the substrate, then the enzyme isn't going to work anymore. So that active site and the substrate have to fit together correctly in order for the reaction to happen. If the wrong substrate comes in, then nothing's going to happen. If the right substrate comes in, but the enzyme shape got changed, the enzyme got damaged somehow and its shape got shifted, then that substrate isn't going to fit the active site, and the enzyme won't be able to do the reaction that it's supposed to do. So it's really, really, really important for enzymes to have a very specific structure, a very specific form to them. So for the most part, enzymes are kind of a little bit delicate. They can only handle minor fluctuations in like temperature or acidity before they don't work as well. So how hot or cold a solution is, or the pH, if it's very, very acidic or it's very, very basic. This can mess with the enzyme's shape. This can break down parts of the enzyme and cause its shape to change. And remember, if an enzyme shape change, it won't be able to do its job anymore. So those are kind of very general environmental conditions that can affect enzymes and cause them to break down. There are also specific structures called inhibitors that can also hurt enzymes in terms of doing the job that they're trying to do. So there are two major examples of inhibitors that I'm going to go over with you guys today. There are competitive inhibitors and non-competitive inhibitors. So each type of inhibitor causes an enzyme to not be able to function correctly anymore, but they do it in slightly different ways. So a competitive inhibitor is a substance that is really, really close to the shape of the substrate that the enzyme normally works with. So because of that, it is able to fit in the active site. It might not be like 100% perfect exact fit, but it's close enough to where the inhibitor is able to bind in the active site. So you've got this inhibitor in your active site. So now your substrate can't get in anymore. Whoops, too far. So it's effectively blocked out. Because the enzyme, it can't do the chemical reaction because it doesn't have the right substrate. But also the inhibitor is able to stick in the active site because it's close enough to the right shape to not be kicked out by the enzyme. So a competitive inhibitor competes for the active site. It takes over the active site and takes it away from the substrate. So now this enzyme is not able to bind to the correct substrate anymore because it's blocked up, essentially. It's like putting the plug on a sink or the tub. Substrate can't get in because the enzyme's all plugged up. It's got this plug in its active site. And we call that plug a competitive inhibitor. The other type also affects the enzyme's active site, but it doesn't do it quite as directly. So if a competitive inhibitor goes in and blocks the active site from the substrate, a non-competitive inhibitor goes somewhere else on the enzyme, but it also messes up the active site. So a non-competitive inhibitor is called this because it is not competing for the active site with the substrate. It's not trying to get in there. But it binds to another part of the enzyme, so some other outside part. And when it does that, it forces the enzyme to like twist a little bit or turn a little bit, which messes up the active site. So now, even though the active site is technically open, it's the wrong shape. The substrate can't bind there anymore because the active site is all messed up. It got twisted and turned around because this non-competitive inhibitor is over here kind of bending the enzyme a little bit. So it's not trying to compete and go into the active site like the competitive, but it is still messing up the enzyme. It messes up the shape, and like I was just talking about, the shape of an enzyme is extremely important. So if you mess up the shape of an enzyme, you break the active site, or you mess up the shape of the active site, and now it can't do its job anymore. The substrate can't bind correctly, and so the enzyme can't cause that reaction to happen quickly. They're both essentially stuck. So competitive inhibitor goes right for the active site and prevents the substrate from getting in there. Non-competitive inhibitor goes somewhere else, changes the structure, which prevents the enzyme from working properly. It stops it from being able to even bind to anything in the first place. It completely changes the shape of your active site over here. All right, does anybody have any questions about inhibitors or enzyme shape and function? Anything that you want me to go back and go over a little bit more?
No, thank you.
So I know that this is a whole bunch of new material that I kind of just threw at you here. Hopefully you liked it. I think enzymes are kind of super cool and interesting. So I'm going to do one kind of quick run through of everything from the beginning, just to kind of review everything over again now that you kind of heard all of the different pieces. Going back. Alrighty. So our enzymes are a type of protein that is a biological catalyst, meaning that it speeds up chemical reactions and makes them happen much, much faster than they would normally. And the cool thing about enzymes is they are not actually part of the reaction. They make it happen, but it only happens in this little area down here, the active site. So the rest of the enzyme out here is not affected at all. They're not changed in the process. It's nice because it makes enzymes reusable. So we have our substrate, we have our enzyme, and we have our active site. And whether or not we've got one big substrate or two smaller substrates, the substrate is going to bind to the active site. The enzyme will then make the reaction happen. It'll go very, very quickly. And then the products, the new structures that we've just made, are going to be released from the enzyme. That active site is open up again, and that means it's open for business, and we can redo this same reaction over and over and over again. Because again, the enzymes are not changed. They're not used up. As soon as the active site is free, we're able to bring in some more substrates and do it all again. So the way that enzymes make all this stuff happen is they lower the activation energy. Essentially just means you need less energy to get the chemical reaction started. Whereas before, you might need this amount of energy in order to get things going. With the enzyme, you only need a little bit of that. You only need a fraction of that original energy. So before, we had to wait until we had this much energy built up before that reaction could start going. With an enzyme, we only have to wait until we have this much energy built up, which can happen much, much, much quicker. Thousands or even millions of times faster with an enzyme versus waiting without an enzyme. So a couple of facts about enzymes. They are very, very specific for the reaction that they make happen. They only can really fit one type of substrate and do one reaction per enzyme. Because of that, we make hundreds of different enzymes for our bodies to do all sorts of different jobs. Enzymes are nice, they're reusable. We can use one enzyme over and over and over and over again, dozens or hundreds or thousands of times before it breaks down. And a lot of times they're kind of named in a similar way. Although to be honest, that's not really all that important for now. So we have our enzyme, we have our little active site opening, and we have our substrate that's going to come in and fit nice and snug in that active site. So our enzyme is this protein that's doing this speeding up of the reaction. Our substrate is our starting material. It's going to fit right into the enzyme and form what's called the enzyme substrate complex, which is just a big fancy way of saying they're both stuck together for a little bit. The substrate is going to bind to the enzyme right at the active site. So the active site is this little pouch or this little hole, this little divot in the side of the enzyme, and the substrate fits right into the active site. So the correct substrate will be able to perfectly fit into the correct active site. If it's not the right substrate for this enzyme, it won't fit and no reaction will happen. So some reactions have a lock and key scenario where the substrate exactly perfectly fits the active site. And then after we make the products, they're a little bit different shape, so they don't exactly fit anymore. So they're kind of let go. They're released by the enzyme. So we have our substrate. It perfectly fits the active site. The reaction happens. We change into the products. And then the products are released. They don't fit anymore, and we want them to leave and go do something else. And then this enzyme can be used all over again. There also is an idea called the Induced Fit Hypothesis, which basically just says that maybe in some cases, instead of the substrate and the enzyme fitting perfectly together, the substrate actually changes the shape of the enzyme a little bit at the active site. And that's what causes the reaction to be able to start going. That's what causes the enzyme to start catalyzing that reaction. So the shape of an enzyme is super duper important to its function. If that active site gets messed up in any way, then the enzyme is effectively broken. It can't be used for its normal purpose or for any purpose, really. They can only do that one reaction. So if they can't do that one reaction anymore, then they are out of luck. So for the most part, they can only function in like certain temperature or certain pH ranges. Otherwise, if things get too hot or too cold for the enzyme, or if they're put somewhere really acidic, then they'll start to break down a little bit and those active sites will be totally messed up. Besides just these kind of external natural factors that are working for enzymes, there's also things called inhibitors. There are competitive inhibitors which compete for the active site. They're trying to get into the active site faster than the substrate, because once they get in there, they stop the substrate from being able to go in and bind to the enzyme. So it's basically like a plug on the active site on the enzyme that doesn't let anything else in. So that's one way to kind of shut down an enzyme from working. Another way is through a non-competitive inhibitor. So like the name suggests, these are not competing for the active site. Instead, they go somewhere else on the enzyme. They go on kind of the back porch over here. And when they bind, when they stick to the enzyme, it twists, it changes its shape. And when we change the shape, that's bad news for the enzyme. Because if the enzyme changes its shape, the active site will change its shape. And now that substrate can't fit anymore. It's no longer able to bind to the active site. So we've stopped the enzyme from working by changing the shape of the active site, by causing the active site to have the wrong shape. If this inhibitor comes off of the enzyme, then it'll revert back to its original shape. The active site will be back to normal and will be able to start catalyzing reactions with that substrate again. All right, and that should be pretty much everything that you need to know for now about enzymes. Does anybody have any final enzyme questions? Anything about the substrates, the active sites, the inhibitors, the activation energy? Any final questions relating to enzymes? Yep, Angelica, definitely a lot of info, but y'all are very smart, so I'm sure you're able to handle it, or at least go back and look at it one more time and kind of have it all sit in your brain. All right. So I knew this would be a lot for today, which is why I made the assignment for today just kind of a simple question to get you started thinking about enzymes. The main work for the enzymes is a worksheet that's going to be posted up tomorrow that pretty much is just a little bit of a review of all the stuff that we covered about enzymes here tonight. So first, there's a little bit of a fill in the blank portion where you're going to use this word bank right here to kind of answer some questions about enzymes, how they are as protein, what they do, what kind of their job is in the cells. There's a labeling section where I want you to tell me which of these labels points to the enzyme, which points to the substrate, and which points to the active site. And then I want you to tell me out of B and C in this picture up here, if we're talking about a lock and key model, which one represents the lock and which one represents the key. And then finally, kind of a final little short answer question about if something that is not the correct substrate enters the active site, what's going to happen? So if you have the wrong shape for something go into the active site of an enzyme, what is going to be the result of that? Okay, so hopefully not too much, only about 7 questions. So pretty simple, straightforward worksheet to hopefully give you some time to start looking at and start working on the midterm for the week. So again, about the midterm, it'll be posted on Wednesday, but you have Wednesday, Thursday, and Friday to work on it if you need to. So take your time, read the questions carefully, go back and look at the notes if you need some help. All right, anybody have any questions about enzymes or the enzyme homework or the midterm before we wrap things up for this evening? Yep, still have class on Thursday. Thursday will be a normal class, just like any other. We're going to talk about some new notes. And make sure to come to class on Thursday if you can, because there are, I think, 1 or 2 questions on the midterm that's information that we'll cover on Thursday. So I think the last couple midterm questions relate to the notes that we're going to go over on Thursday. So I would definitely recommend being there if you are able to.